everybody. Welcome to the Research Minute Podcast. I'm your host, David Butler. This podcast takes university research from the campus and brings it into the community. Today on the program, we have Kevin Bicker, professor of chemistry at Middle Tennessee State University. Welcome to the program, Kevin.
Thanks, David. Great to be here.
Well, appreciate you being here. We've worked together for a wee bit of time.
We bit.
You're a chemist.
I am.
So you study chemical things, these molecules and all
Sure.
this stuff that everybody loves taking classes on.
Yeah.
When did you know you want to be a chemist?
In college. So I started as a pre-medicine. I wanted to get into surgery, be an orthopedic surgeon.
Not just I want to be a doctor. I want to be an orthopedic surgeon.
I wanted to be the construction worker of the medical field. It was,
Wow.
It looked amazing to me.
How did you know? Not only do you want to go get an MD, but then you wanted to do that extra 3 to 5 years past residency. Why orthopedics?
I had a lot of experience with orthopedic surgeons. I was an athlete, injuries, seeing the doctor all the time, and I enjoyed the sort of work they were doing. So I connected with a surgeon. I sat in on some surgeries. I shadowed what he was doing, and it was fascinating to me. And so that was the plan. And then when I got into college, advisor.
So you did all this before you even went to college?
Yeah, when I was in high school, junior and senior in high school.
You're like sitting around and they're like, let me build a new knee here. Let me do a shoulder.
Yeah, I sat in on my mom's carpal tunnel surgery. It was, yeah, it was great. But then I got into college and I started taking some different classes. Advisor said, hey, why don't you double major in biology and chemistry? Because then you have all the electives you need. But you can go do lots of different things if you change your mind.
Right. A lot of flexibility.
So I did.
And you were doing pre-med. So what was pre-med? Only biology at the time?
Pre-med is a mixture of both biology and chemistry.
Okay. And where were you? Where were you doing your undergraduate?
I was at Bluffton University, tiny little liberal arts school up in Ohio.
And where in Ohio?
Western Ohio, sort of middle of nowhere. North of Dayton, south of Toledo.
Western Ohio, but in the middle of nowhere. Is that the tagline for the thing?
I think that's the tagline for most of Western Ohio, probably.
That's good. We didn't go far enough to get into the other state, but we're still in Ohio.
That's right.
Okay. All right. So you sat through your mom's carpal tunnel. I'm pre-med. You're going to take chemistry and biology classes.
And then I got into organic chemistry, and I fell in love, which doesn't happen for a lot of people.
No, because most people avoid the carbon chain.
They do, but it just clicked. I enjoyed it, and I had some great professors, and as I got more into that world, I began to realize that maybe that was the career for me, that I really wanted to be a professor and teach.
As an undergraduate, you're like,
yep.
Okay, this is really strange, because you're like, I'm in high school. What are you going to do? Oh, I don't know. I'm going to go to college. I'm going to be an orthopedic surgeon.
Yeah.
Now here you are doing pre-med. You take organic chemistry, you're like, oh, I'm going to be a professor.
Yeah, I'm always pretty definitive on what I want to do. I'm usually wrong on how that turns out.
But you're definitive about the pathway at any given time.
That's, typically. So I went to graduate school at the University of South Carolina for chemistry with a focus in organic chemistry.
And this is in Columbia, right?
And this is in Columbia, South Carolina. Yep.
And that's in the middle of nowhere on the west side of something?
Have you been to Columbia?
Yes.
It's hot and humid.
And humid. There you go.
I usually tell people it's like living in a dishwasher.
Yes.
Living in Columbia. But it was good. It was a good school. It was a good experience. And it taught me a lot. And one of the things it taught me was that I enjoyed research.
Okay.
And I still loved teaching. But research became something that was enjoyable. And in terms of the output of research, I was starting to get into biomedical, bioorganic, whatever you want to call it. And I began to see that my research could have an impact in the medical field, which is what I wanted to do originally.
Right, because that medicine leaning and that surgery piece.
Yeah, but it would be at the really early end of that. So wrapped up graduate school, I worked on peptide libraries at that time. And
so let's explain what a peptide is.
Peptide.
And that it's not sitting in a library with books. You're not pulling it off the shelf.
No.
A library is a what of a peptide?
Yeah, so a library is a collection of molecules. Like a library is a collection of books. In chemistry, a library is a collection of molecules that are all kind of similar. And a peptide would be this little molecule. We find them all over nature, and they serve a lot of different purposes in our body and out in nature. And so that's what I was working with. And we were trying to come up with treatments for cancer or diagnostics for cancer. And that's ultimately where we landed. We developed a technique that allowed us to determine how aggressive a cancer was, And that's really where that project sort of landed.
So
by
using
the end.
peptides as a mechanism for early screening to determine if cancer's there and then how aggressive it is.
That's right. Finished up there and moved down to South Florida to take a postdoc position at the Scripps Research Institute, working there in a more of a biochemistry lab.
Okay.
So taking my organic training and taking that into a biochemistry lab to learn some new skills. I worked on enzymology, which is the study of enzymes, which are these proteins that can do different functions in our bodies. We were really interested in some enzymes that were related to different inflammation and autoimmune diseases in the body. And so I was working on developing molecules that could block the activity of those enzymes. That research was great. It taught me a lot on the biochemistry and biology side of things. And so then I came to MTSU and started my own lab. We started working on trying to develop anti-infective peptoids.
Okay. So what's anti-infective? Let's start with that.
Fighting against infections caused by things like bacteria or fungus or a virus. So when you get a cold, you get ear infection, you get COVID, those are infections. And so we're trying to come up with molecules that can fight against those infections.
But don't we have white blood cells that kind of come in there and do that already?
We do, and they're doing their best. But, some infections are just too severe, aggressive, or evade the immune system and can get pretty serious.
Okay, so peptide we know is this molecule that exists in nature and everything else. You're using it as diagnostic for cancer, et cetera. Now, what is a peptoid compared to a peptide?
Yeah, so we threw in a new little word there. So a peptoid, which has been around for 30-ish years, it looks, if you saw a picture of it, the chemical structure, it would look a lot like a peptide.
Okay.
There's just this little change in the structure.
Okay.
And the benefit is that it stays around a long time in the body.
Okay.
So
compared to like a peptide?
Compared to a peptide.
So a peptide lasts about how long in the body?
So what we measure is something called half-life. And so that's sort of how quickly it decays, cuts in half, cuts in half, cuts in half. And so a typical peptide, 10 to 15 minutes.
Okay, wow, that's fast. Peptoid, longer than 15 minutes.
About 24 hours.
Okay, typically. That's significantly. Doesn't sound like very long. I mean, one day, but compared to 15 minutes, that's a huge multiplier.
That's right. Right. And so it allows for longer for that molecule to find the bad bacteria or fungi, kill it, and do its job.
Okay. And so there are certain medicines right now, pharmaceuticals, that use peptides that go into your body but can't hang out as long and can't fight as long?
There are some. Nowadays everybody's pretty familiar with something like Ozempic.
Yes.
Right? So Ozempic is a peptide. And they have done some things to Ozempic that it lasts longer. So it has a better half-life for sure. So in chemistry, we can mass produce these chemical libraries. Okay. Where in a matter of a day or two or a week, we can synthesize 1,000 to maybe even 100,000 molecules in the matter of a few days.
And these are the identical molecules, or they're changed a little bit because there's a carbon here, not a carbon here, or something switched up? Is same structure, or just a little variation on each?
Little variations.
Okay.
I sometimes use the analogy of Legos. I know you're a big Lego fan. Yes. The way that we make them really is like stacking Legos on top of each other. So if you built this Lego tower, you build it one time and you use different colors, right? And the next time you build it, you use slightly different colors. And so if we had a library, it would be like there are a bunch of Lego towers, but each one is a slightly different combination of those different colors of Legos, where each color for us is a different chemical group. And so we can pretty rapidly just produce these huge collections. And that's actually the easy part. The hard part is then trying to pick through that library and find the ones that really have good activity, the ability to kill the bad stuff that you want them to kill.
Production of large volume, easy. Then right now you've got to put it to work.
That's right. That's right. Yeah, we typically use the phrase interrogation, right? Sounds like a cop show. Yeah, but we're taking this.
You waterboard the molecules until they give up their properties?
No, it's organic chemistry, no water.
No water. Hydrophobic.
That's right. The specialty that my lab has gotten into is developing techniques that allow us to really quickly screen or interrogate these collections of compounds, these libraries, and find ones that have good activity against the bad bugs that we want to kill.
Okay. So these peptoids, you're building them easily.
Yep.
But then you've got to test each.
Traditionally, you would synthesize each of those molecules one at a time.
Right.
Clean them up so they're nice and pure.
Okay.
And confirm that they're structured.
All the Legos in the right place.
All the Legos are in the right place.
Okay.
And then you would test them one at a time against these bacteria and fungi. Very time consuming.
Yes, it sounds like it.
So that's where we came in, because my training is specialized in what we call high-throughput screening.
Okay.
Screening or interrogating these libraries in a high throughput or quick way. When you look at the typical pipeline for developing a drug molecule, going from initial development or initial discovery through clinical candidate.
Yeah. And that means through people.
That's right.
People.
10 to 15 years.
Right.
Billions of dollars.
Yes. Literally billions of dollars.
Billions and billions of dollars.
Each stage costs in the millions of dollars.
That's
people
right.
and samples and production and safety.
But that cost and timeline is not linear. There is a lot of cost and timeline on the front end. Okay. Because you have to get through thousands and thousands of individual compounds before you find the ones.
Down select to that single one.
That's
the
right.
magic bullet.
That's right. So anything you can do in that early phase to speed that up and make it more cost efficient accelerates dramatically everything else.
And decreases costs.
And decreases costs.
So it's speed and time money savings if you can figure out that log jam early on.
That's right.
Okay.
That's right. We do this high throughput screening, right? So rapidly interrogating all these molecules at one time. The way that we do it is we take a Petri dish and we take some of our library, which they're attached to little beads.
Okay.
So imagine a bead that's like a 0.5 millimeter in diameter. So it's teeny tiny.
Yeah.
You can see it, but it's real tiny.
Smaller than a regular BB.
Yeah. And each one of those has its own peptoid on it.
Okay.
So if we say we have 10,000 peptoids, we have 10,000 of those little beads. And each one has its own peptoid.
Okay.
And on there it's going to have thousands of copies of that peptoid, but just one type of Lego stack. So we take some of that library, we put it into a growth media that the bacteria can grow in that's been liquefied. Okay. And then we put it into a Petri dish and we allow it to solidify. Okay. And also in that liquid media is the bad bug that we want to kill. Right. All right. So we allow that to solidify. So now what we have in our Petri dish is solid media filled with bad bacteria, and embedded in it are hundreds of these little beads. Yep. And we have some ways, I won't go into the technical details, that allow us to release some of that peptoid from the bead while keeping some of the peptoid on the bead. Okay. And so some of that peptoid comes off the bead, it goes into that surrounding growth media where the bacteria is and interacts with the bacteria. It's
like a slow release.
That's right. And if the peptoid can kill the bacteria, then overnight we see this like nice lawn of bacteria, but there are these little moon craters in it.
Like Roundup on your grass.
That's right. And so that's where there was a peptoid that was released that is killing that bacteria. So let's say we've done this screening. We screened thousands of compounds, and we ended up with 30 leads. We're going to take those 30 leads, we're going to remake all of them, and then we're going to do that traditional testing. Okay. And that's going to allow us to know not just do they kill the bacteria, but like at what concentration. Okay. Right? And we want the lowest concentrations possible. That way we're using the smallest amount of molecules we can.
I gotcha.
To kill that bad bacteria. And what we're looking for is a window between the amount we have to use to kill the bad bacteria or fungus and the amount that that is toxic to those mammalian cells.
Okay. So you're looking for a minimum common denominator, but you also need to know what the maximum strength is before you hurt somebody.
That's right. That's right. And we just want that window to be as big as possible. Because like you said, if that window is really big, then as we start getting into animals or humans, we can dose that up and we have room to do that without causing toxicity.
So not only will it last hopefully 24 hours in the body, but you can give them 10,000 of them because it takes 10 million before they die.
That's right. So that's sort of our initial phase of that pipeline. Okay. That usually rules out maybe half of the compounds. So we're going to move those to the side. And then we're going to take, let's say, those 10 compounds, and we're going to move those forward into more detailed testing. Maybe we test against other bad bacteria and fungi. because, infections that people get, they're not always just one bad microbe.
Right.
They can be combinations.
Right.
So we want to see, does this thing kill multiple bad bugs, which would be really valuable.
Yes.
Once we've done that optimization, let's say we have at that point maybe 3 really high quality molecules.
Right.
And so now we're going to take those forward into what we would call therapeutic characterization.
Okay.
Is it synergistic? So if we use it along with existing antifungals or existing antibacterials, does that lower the amount that we need to use?
Right.
Right. Which
So you don't want that toxicity piece.
That's right.
And something that already exists on the shelf.
That's right. That's right. So we're testing things like that.
Okay. Interesting.
Yeah.
So what's interesting to me is you have to discard 95%, maybe 98% of a lot of these unique peptoids.
Yeah, for sure.
But the good news is you can synthesize 50,000 per day.
That's right. We can design libraries that have billions of theoretical compounds. We can't synthesize a billion compounds.
Right.
We're limited logistically. Max we could synthesize is maybe a couple 100,000 at a time. But in terms of the chemical space that we could sample, it's massive.
And let's make it clear to the audience, this high throughput piece is a technology you invented.
That's correct.
That has a patent to it. So this is not something that every pharmaceutical company, not Merck and the Johnson, these people do not have this in their laboratories. No other university has this in their laboratory. It is in the Bicker lab in the Department of Chemistry at Middle Tennessee State University only.
That's right. Yep, that's right. Yeah.
This technology has an opportunity just to transform the idea of using these peptoids and getting them out to market in a way that I just couldn't imagine laboriously trying to measure these over time.
Yeah, I certainly hope so. There are a handful of people around the world trying to develop peptoids as anti-infectives, and we desperately need new anti-infectives. You mentioned a couple of big pharmaceutical companies there. They don't research antibacterial and antifungal drugs anymore. The profit margins are too narrow.
Right.
So we need labs at places like MTSU trying to identify these new medicines because bacteria and fungi are not going away.
No, they seem to want to adapt. Every time we do something, they seem to counterpunch us back.
It's an arms race.
Yes. Yeah. Project for me in the next 3, 5, 10 years where you think this peptoid research, the assay, this pipeline may go.
We sort of have things in different phases of development.
Okay.
We will continue to discover new leads to identify new bacterial or fungal targets that we want to start developing peptoids against and just keep working through those. We also have leads that are in those later stages of development. And so we're going to keep pushing those forward. When we reach the end of what happens in my lab, right, we go from design, synthesis, discovery, validation, and development.
Right.
When we reach the end of that pipeline, it's time to get into animal models.
Okay. And animal models meaning taking that drug and putting it into a live animal.
That's right. And, looking at how long does the drug last in the animal, or if the animal has an infection, does the molecule combat that infection? That's not something my lab or my training, I'm not set up to do that.
Right.
So we collaborate, we partner. We have, as you know, been developing a relationship with an entrepreneur to launch a company called Pepsana. And that company is solely focused on taking the peptoids that my lab develops and trying to push those towards commercial and clinical use. Okay. And so we've been doing that through applying for grants, trying to secure capital, but then also developing partnerships with the federal government, private industry, the U.S. Army, to test against really nasty things like anthrax. If we were to have this conversation 5 years from now, what I'm hoping is that we have some of those leads out in trials in animals or progressing towards clinical trials in people. The new leads that we're trying to discover right now, we're getting ready to launch into those animal studies just to keep moving things forward towards that clinical application to really deliver what people need, which is new medicines to treat really nasty infections.
Okay, well, great. Well, thank you for taking the time and joining me today.
Thanks for having me, David. Great to be here.
I want to thank Kevin for joining us today on the Research Minute podcast. And remember, there's a peptoid in your future.